Well, hello there, friends. Um, Been a while. Sorry. Got caught up in exams in nursing micro and doing different things. But I actually have a couple of ideas cooking that next week we'll be doing a beginnings of our first sort of in vitro lab or virtual lab. I'm going to give you some sequence. We're going to make some primers and we're going to see what kind of PCR products we get. It's one of the things that doing the virtual form of molecular genetics is so perfect because everything works exactly, whereas the reality is oftentimes a lot more confusing. But when we were last talking about genome complexity and doing things like COT analysis and the recognition that the genome is comprised of unique elements, which are typically messenger RNA coding genes, and then moderately repetitive sequences, things like ribosomal RNA, tRNAs, and then highly repetitive sequences, which are these very short sequences that are either repeat sequences or very simple repeat sequences that like 3 or 4 nucleotides links repeats, or sometimes they're a little bit bigger. Some of these make up actually the DNA sequences that are used for CODIS DNA analysis. I thought what we would do is today talk a little bit about some of the techniques behind DNA sequencing, because what we're going to be working on will eventually result in required DNA sequencing. So DNA sequencing is a really critical tool, and those of you that were in Molec and Cell, you perhaps remember our discussion last spring with members of the University of Illinois Biotechnology Center, and they were talking about a variety of different sequencing systems that they use. So one of the things is that DNA sequencing technology advances very rapidly. We're going to kind of talk about some of the more standard systems and actually kind of things that we will be using. So DNA sequencing begins really with some of the initial analyses of DNA sequence and the development of tools to be able to actually identify individual nucleotides. The first real sequencing work, the sequencing protocol, was developed by two guys, Maxim and Gilbert. And this is the original DNA sequencing technology. Maxim and Gilbert published a 25 nucleotide sequence in the Proceedings of the National Academy of Science of the lac operon. It was the very first DNA sequence ever published. And it was like 25 nucleotides. Whoopee! We now talk about routinely doing 800,000 nucleotides in a sequencing run. But this was a very significant event. Now, Maxim and Gilbert sequencing is a fairly difficult thing to do, and it principle that will be actually in terms of experiments be using a lot, in that we're talking about, at least in other experimental systems, is that you take a double-stranded DNA molecule and you end label one end using either radioactively labeled nucleotides that you can fill in one spot, or enzymes that will actually attach a radioactive phosphorus to one end. The principle behind this, though, is this. You end up with a radioactively tagged DNA. So it's on one end. And then what they do is they subject this DNA to a range of different chemicals, including hydrazine, which is actually used as rocket fuel, and different kinds of conditions to break bonds between the A, T, G, C, different individual nucleotides. And the specifics of it aren't that critical, but you end up with bonds that are having breaks between A and T, or breaks between A and anything else. And then you have breaks at G's and breaks at C's. And you end up with a series of fragments that'll look like this. So you'll have, say, your G sample that you're going to be breaking at individual G nucleotides. And what you do is you have the conditions set up that you generate. So let's just say there's a G here, and a G here, and a G right there. All right. You generate molecules that'll look like this. And another one that'll look like that. Here's the radioactive tag end. And then another one that'll look like this. All right. Now, if we think about this, we also can generate, if we're breaking apart the strands at any G, you can generate a fragment that'll be there, or a fragment that'll be there, or a fragment that'll be there. But the only fragments that we're going to be able to visualize are those that are carrying the radioactive phosphorus at the end. And so these internal fragments you don't see. Now, what this means is you can prepare the 4 nucleotide fragmentation systems, breaking either at A's or between A and T and G and C. And from this, we can then separate these fragments out on a gel. Oop, this marker is dead. So, on a gel, doing acrylamide gel electrophoresis. And the gels end up being really, really big in order to have sufficient resolution. I mean, literally, the gels would be the size of a window pane, or a full glass chunk of window, big glass. Very, very thin, so they have a very high resistance. It generates a lot of heat. And what you do is run the DNA out, and the fragments that are with the radioactive end, those are the ones you'll see when you do an autoradiograph. You're going to take this thing, the gel, and actually put it onto a sheet of X-ray film. And when you develop the X-ray film, you'd see something that looks like this. So you have this band being small will be down here, this one will be up here, and this one would be right there. All right, because the smallest is down here, and then these two are right next to each other. And you do the same with the other nucleotides and with the other nucleotide preps. So the Maxim-Gilbert system requires an end label, and you're breaking using a series of different kinds of chemical reactions, very harsh chemical reactions to break this thing apart. It requires a lot of DNA, and it is a pretty ugly and inefficient system. And the ability to generate a lot of sequence is quite limited. So this is the technology that was being used for a while, but by the time we get into the late 70s, early 80s, a new technique comes about. And this was developed by a guy named Sanger. And Sanger sequencing is a much more efficient, much more interesting system. That's an S there. Sanger sequencing uses a modified nucleotide that we actually made allusion to when we started talking about DNA topology in the 5' end, 3' end. Sanger sequencing uses something called a dideoxynucleotide, and it's often referred to as a chain terminator. Meaning that when you incorporate a dideoxynucleotide, you're going to block any further DNA polymerization. So let's look at what a dideoxynucleotide looks like. So we've got the 5' carbon up here, the nucleotide identifier base over here, which really doesn't have anything to do with our work, and then we've got the hydroxyl over here, or if this is DNA, it may be deoxygenated and just be a hydrogen. And here, at the 3' carbon, we have the OH. Now, if you recall, this 3' carbon is critical because we're going to make the phosphodiester backbone link. When we take the next nucleotide, we're going to take the phosphate molecule and attach that here with the through an oxygen. That's critical. And this is how we carry out DNA polymerization. So the 3' hydroxyl is critical for making the link of adding the next nucleotide. If you incorporate a di-deoxynucleotide with a hydrogen here, nothing happens. You can't add a new nucleotide here, so it stops in its tracks DNA polymerization. Now, rather than using chemicals to break apart the nucleotides, Sanger sequencing is using DNA polymerase and a mix of standard deoxynucleotides and then a very small amount of a dideoxynucleotide. So the original way you would do a Sanger DNA sequencing reaction is like this. You take, and there's a couple of different ways you can play with this. The easiest way to do this or think about this is you take your double-strand DNA molecule and we're going to heat it up so it's single-stranded. We have to have a little primer sequence. And so we got single-strand DNA and we're going to add a little primer sequence, 5' to 3', and we're going to begin putting in nucleotides. All right, so that's a standard DNA polymerization reaction. There you go. Now, for DNA sequencing, what we do is we initially throw in radioactive phosphorus in the form of P32 DCTP, just as a general one, and that begins to incorporate at every C a radioactive nucleotide. All right, now you do this under conditions that you don't allow for too much polymerization. Typically what you're doing is you have a limiting amount of the nucleotides, but no dideoxys. You just let this run for just a few minutes, 10 minutes or so, and that's going to label up the DNA a little bit. Next, you take this whole thing and you throw it in to 4 separate tubes. You just split out your DNA that's been labeled a little bit into 4 separate tubes. You have the A tube, the T tube, the G tube, and the C tube. Okay, so a little bit of this, 1/4, goes into each one of these tubes. These tubes are set that they have, all of them are going to have DNA polymerase. They're going to have all 4 deoxynucleotide triphosphates, DNTPs. And then, depending, they're going to have 1 dideoxynucleotide triphosphate. And the amount of dideoxy has to be calculated and kind of tweaked a bit. You don't want to have so much dideoxy that you can never get past the first nucleotide of whatever that nucleotide is. You want to have a little bit in there. I mean, it's a little bit like, so, you got all 4, but then you also have the dideoxy. So you kind of reach into the bag and you pull out a marble. Most of the marbles are going to be white, but occasionally you're going to pull out a red one. That's going to be the dideoxy. All right, so the way this works is the bulk of the nucleotide or fragments are going to start to be kind of short, but just because the odds of being able to go a really long distance are kind of hard, because each time you reach in there, you have a chance of pulling out a red nucleotide. But those that are bigger actually incorporate more radioactive nucleotide, and so they have a stronger signal. And so you end up with a range of fragments now, a whole different size range, and you run these again out on a gel. So you have ATGC, and the gel will look like this. So you'll set it up pretty much just like this, where, I mean, the lanes are a lot narrower, but this gives us the idea, in that we have ATGC. You have the A reaction, the T reaction, the G reaction, the C reaction. And you'll end up with a set of bands, and I'm just sort of messing with this here. And what you have here is a ladder, and you read it from the bottom up. And so the sequence here would be A, A, T, G, C, 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 G, T, A. And if you're good, you might be able to get, between 150 nucleotides, maybe 300 nucleotides. Part of the problem is that as the gel runs, the way electrophoresis works is that the distance migrated is not linear. It is a logarithmic function, and stuff separates out bigger down here than it does up there, and you end up with a gel that has a lot of bands compressed at the top. I'm just going to actually pull up a picture here of a DNA sequencing gel, and let's see. You can find all kinds of sequencing gels as well, but let me very quickly just show you one. So this is just an example of what a sequencing gel might look like. And you very carefully just start at the bottom and start working your way up to the top, reading the sequence. So we're able to generate a range of sequence. This is a fairly difficult process. You got to have a lot of radioactive nucleotides floating around, so it's radioactive. The gels are not easy to make. And you're limited by how much samples you can read by how far the gel can run. So by the middle 80s, late 80s, technologies and chemistries were being developed to shift this away from being using radioactive tags to developing a fluorescent tag. And it's actually a pretty cool little trick. So we're going to use the exact same kind of mix now using dideoxys. But instead of using radioactive label, chemistries began to be developed that would generate a fluorescent tag on the nucleotides, a fluor. And there were two competing technologies, one from a company called Spectrophysics, who developed a chemistry where they had a primer that had a fluorescent tag. And then there was another company, ABI, that developed a chemistry that they had the dideoxys individually labeled with slightly different color fluorescent tags. And from here, you can now shift this into a different kind of process. So we've got different fluorescent tags here, and just, we can kind of pretend you have the dideoxy is labeled with either a green fluor or a blue fluor or a black fluor, well, whatever, and a red fluor. Red's not working so good. With a red fluor. All right. So now, rather than having to run 4 separate tubes, you can run this all in the same tube using these 4 separately fluorescently tagged nucleotides. Now, the colors that we generate are rather different. The actual color difference in the fluorescence is not all that different. You couldn't see it. And so now, instead of having to run this on acrylamide gel, which initially they did, but fairly quickly, a new technology using a capillary tube is something known creatively as capillary electrophoresis is the tool. And with capillary electrophoresis, we've got, as the name implies, a capillary tube, not nearly this big, the size of a little coffee stirrer, packed with a resin. And it still is going to migrate with an electric field. You put the sample on top, the individual fragments come out the bottom. And as they come out, there's a little laser scanner that is looking at the fluorescence emission spectrum of everything that drops out. And you get, now instead of an autoradiogram, you get something that is known in the biz as an electrophoreinogram. And this is the kind of tool you can use to actually begin now looking at pretty long amounts of sequence. So you're not trying to look at resolution on a gel. You're just running this through a capillary and looking at the very bottom. And it's not uncommon to actually have 300 or 400, 800 nucleotides in one run. So I'll just show you what that looks like. And this is typically what our sequences look like that we use now. So this is a foreinogram. And you have these peaks. So when we get back some sequence data, which I hope we will, from DNA that we're going to amplify, we'll be able to go through it and see what it looks like. What this allows is for automated DNA sequencing. And that leads us into the tools that we're going to need to be able to actually do large-scale genome projects, which we will pick up on in our next discussion. All right, so I have not forgotten about you. Do not worry. We're going to put together a little exam type of thing that's going to be a take-home or do at home, no time limit. I'll have that out for you probably for next week on some of the stuff we've been covering. And we'll see where we go from here. All right, thank you for your attention. Don't be a stranger. I'm here. And I'll check in with you guys later.